This method can help answer key questions in the molecular biology and genetics fields of Hymenoptera, such as sex determination systems. The main advantage of this technique is that we can easily induce inbreeding lines of the ant Vollenhovia emeryi, which are essential for genetic mapping and molecular studies.
Though this method can provide insight into the sex determination system of a specific ant, it may also be applied to other ants and other Hymenopteran species, such as wasps.
To begin the protocol, collect V. Emeryi colonies during early summer. Transfer the ant specimens from the collected branches to an artificial plaster nest with a glass cover using an aspirator. Maintain the colonies in the artificial nest at 25 degrees C under a 16 to 8 hour light-dark cycle. Provide tap water with a wash bottle to keep the plaster moist every other day at the same time. Next, add about 100 micrograms of dry cricket powder wrapped in aluminum foil and a brown sugar water-filled tip to the colony, using a 20 microliter tip every other day until new reproductive ants emerge.
It is important to collect mature colonies from the fields and to provide them enough food to gain new queens and males.
First, stop individuals from moving by placing colonies in a room with a controlled environment set at 4 degrees Celsius for 15 minutes. Remove the mid-legs of 30 worker ants using forceps under a stereoscope to distinguish the F0 generation from the workers produced in subsequent inbreeding crosses. Then transfer the ants into a new smaller plaster nest for inbreeding crosses.